Hello everyone, I'm Alejandro Montenegro and I'm the executive editor for Cold Spring Harbor Protocols. I'm joined today by Arturo Álvarez-Buylla, who is a professor at the University of California, San Francisco, to talk a little bit about the research and the work they'll present at the 87th Cold Spring Harbor Symposium, which this year focuses on stem cells. Thank you very much for being with me today.
Pleasure.
So research in your lab is centered around neuronal origin, proliferation, migration, and integration in the adult brain. So tell us a bit about that and what you mean by integration.
Well, I think the ultimate task of a new neuron in the environment of an adult brain is to get integrated into the circuits and to contribute to these circuits. So cells are heavily wired into many, many circuits within the brain, and depending on the neuron, they are more or less interconnected. And this connection is what underlies brain function. Mm-hmm. So when you have neurodegeneration and with the hope of one day, replacing some of these neurons that are lost, one of the tasks that we have to accomplish is have these cells become rewired in a way that is functional and contributes to the repair. Mm-hmm. So the fact that some of these processes that were thought to happen only during development can in fact happen in adulthood. Mm-hmm. And it's not only the last step that you mentioned, integration, but the migration is a very fundamental process because the cells have to intercalate to get to the right places to eventually repair a broken circuit. Mm-hmm. These processes can occur with certain types of neurons in the environment of the adult brain. Mm-hmm.
Mm-hmm. In your website and part of your research, you describe that neural stem cells and related cell types are, like you mentioned, are present in the adult mammalian brain in the ventricular subventricular zone. Can you tell us a little bit about this zone and about the role of these stem cells in the adult brain?
Well, about 20 years ago, this was a huge surprise that the adult brain retained, at least in some mammalian species, cells that have the potential to form new nerve cells and also glial cells. Though people knew that glial cells are the support cells in the brain, had some ability to divide and reform, it was not known that nerve cells could do it. And this began changing in the 60s with some reports from Joseph Allman suggesting that there were parts of the adult mammalian brain that received new neurons. Mm-hmm. And then there was extensive studies in songbirds that demonstrated that new neurons could be made. So a search began for the primary progenitors of these new neurons, what we call now the neural stem cells. And it was very controversial initially, but these cells turned out to be a subpopulation of cells that had been identified previously as astrocytes that were thought to be in the glial lineage. And that has changed conceptually how we think about the lineage of brain development, where the main trunk of where the cells come from actually come from cells that were initially thought to be glial cells or astroglial cells. So these are what we call now neural stem cells, mm-hmm. and the place where they are most abundant happens to be this area of the brain we call the ventricular subventricular zone. That is nothing more than the walls of the big cavities inside the forebrain, the lateral ventricles, and on those walls is where these cells sit, and the niche that supports them to produce new neurons is also there. So the whole machinery for neuronal generation is precisely in these areas of the brain on the walls of the lateral ventricles. This is very, very abundant in the young adult mice. The organization of the human subventricular zone is slightly different, and mm-hmm. we're actively studying it, but I don't think there is active neurogenesis going on there in adult humans. Unlike mouse, that some level of neurogenesis continues into quite old age, although very diminished compared to what you see in younger adults.
Mm-hmm. So, related to that, so you mentioned that this neuronal replacement continues postnatally in some mammalian species, and this happens mostly in the olfactory bulb. So, is it known why is the olfactory bulb the place where this happens and not other regions?
Well, there's been multiple hypotheses raised, and I think one of them is actually receiving a lot of support. So the interesting thing is that the olfactory bulb is at a distance of this wall of the lateral ventricle where I told you, and one of the huge migrations for these cells happens precisely in this pathway from the place where the cells are born on the walls of the lateral ventricle to go all the way to the olfactory bulb. Mm-hmm. So this is a big flow of neurons through a pathway called the rostral migratory stream, through a very specific form of migration called chain migration that moves cells quite quickly in the mouse or in rodents from the walls of the ventricle all the way to the olfactory bulb. Once they get into the olfactory bulb, they become integrated into two main cell types: granular cells that are local circuit interneurons that regulate the activity of mitral cells that are the big output neurons in the olfactory bulb, and periglomerular cells, another set of local circuit neurons that regulate the input of signals coming from the receptors, the olfactory receptors that receive the odors in the environment. So people believe that this is actually a form of constant plasticity to adapt the olfactory bulb to have the best possible perception of odors in the environment. So it's creating a custom circuit for environment that's changing. So as the environment changes with the seasons, for example, you need to have the olfactory system tuned to the best possible reception of the odors that are relevant to the animal. So olfaction is a fundamental sensory system for many mammalian species, and in these mammalian species, this process of adult neurogenesis is well manifested.
So in your abstract, you mentioned that you will discuss findings suggesting the presence of a relay subpopulation of primary precursors in adult brains. What do you mean by this relay?
So this came about because based on recent work from a postdoc in the lab, we realized that these cells were not constantly self-renewing like we had assumed, and that there was a constant supply of these neurons for good in the life of the animal. So that's how it happens in other systems where there's stem cells, but in the case of the brain, these cells seem to be dividing symmetrically most of the time. And when they divide symmetrically to generate progeny, which happens 80% of the time, as we have calculated, those cells are gone. So you start consuming this population of cells. So when we counted the number of cells that we had in the animal, we saw that these cells were progressively decreasing, actually very similar to what the model of symmetrical division suggested, the 20% ratio I mentioned to you. There's a small number of about 20% of these cells that seem to self-renew and produce another cell that has the properties of these primary progenitors and is capable of generating neurons also, but we do not know for how long that can go. The fact is that these cells were coming down. What was a big surprise is that when we measure the number of new neurons, they were not coming down. They also come down, but they were not coming down as fast as the decrease in the number of stem cells. So we began looking with the work that of a postdoc in the lab, Arantxa Cebrian, for possible other progenitors within these walls of the ventricle. And there's a cell that we described many, many years ago. We call it B2 cell. That's another astroglial-like cell that is there. That's a little deeper within the wall. And the work has mainly shown that these cells can actually work as progenitors for new neurons also. So it's like a relay, like passing the baton from the primary progenitors. And we also have some evidence that the cells that touch the ventricle are actually giving rise to these secondary stem cells. So you can see that actually there's heterogeneity among these stem cells, both in space and time, because something I didn't mention is that the reason why this migration happens is that the subventricular zone is organized like a mosaic, and different stem cells in different subregions are specialized for producing different types of neurons destined for the olfactory bulb, because there's a diversity of neurons that get integrated into the olfactory bulb. So the reason why you need to import them from so many areas is that the factories specialized for the production of these different subtypes of neurons are within these different regions. So this relay of B1 to B2 cells happen in each one of these regions. And in some regions, actually, it happens very early, and all you have is B2 cells for the rest of your life.
How do these findings relate to brain repair and plasticity in the postnatal brain?
So these are two different questions. And as I mentioned before, the ultimate goal of repairing brain when neurons are lost would involve many of these processes that happen naturally within two regions of the rodent brain, the olfactory bulb and the hippocampus. So studying deeply what are the mechanisms might allow us to induce them one day with other populations of neurons. This is already happening to some degree with populations of neurons that naturally can migrate within the brain. So based on this study, other studies, mostly with collaborators at UCSF, had led to the identification of cell types that can migrate anywhere in the adult forebrain. And these cells become local circuit interneurons that might be helpful one day for disease. So the whole idea that you can have new neurons integrating, even if that process specifically does not happen naturally in the adult human brain, is inspiring new research that suggests that you can actually have the process of birth, migration, and integration, and especially this latter two, the migration and integration, would be actually fantastic in the case where you have diffusely lost a group of neurons and that are causing imbalances in those circuits. So this is, a big inspiration for our lab and our groups at UCSF to try to move some of these findings towards, applications. In terms of plasticity, it's perhaps even as fascinating a problem as is the problem of repair, because it kind of suggests that perhaps certain kinds of circuits, and I can discuss why the olfactory bulb might be one of them and the hippocampus another, might require this extreme form of neuronal replacement to have enough plasticity, because obviously there is plasticity in many parts of the brain that do not incorporate new neurons. And the incorporation of new neurons is costly because you need a progenitor, you need migration, you need integration. But the other thing is that it takes time for all of these processes to come in, and many times the plasticity needs to be almost immediate. Mm-hmm. So many parts of the brain can function without this form of plasticity where you replace the whole neuron. But there might be cases in which the fixed nature of the anatomy of a neuron, with its dendritic projections and axonal extensions, make it incompatible with forming a new circuit. So you might be able to change the strengths of those connections, but you might not be able to really change the nature of that circuit. And in those cases, having this form of plasticity might bring you back to earlier stages of development where this happens. In fact, we know, in collaboration with other groups at UCSF, that we can induce plasticity by transplanting young neurons into older parts of the brain, specifically local circuit interneurons that are able to somehow change the nature of the circuit within that region. We don't know exactly how this happens. It's something we're trying to investigate, because there is new integration of neurons in that area. Cells migrate in, integrate, and become part of those circuits. We assume that creates the ability to open a new period of plasticity. So for many parts of the brain, these periods of plasticity open and close, and then they're closed for the rest of your life, and the type of plasticity you have is very limited. That's why it's so easy to learn a language as a kid, but then it's much more complicated. So, supposedly this is because of the age of the neurons and how fixed they are within those circuits. So having new neurons can probably open the ability of the brain to readapt to information that might not be able to process otherwise.
Mm-hmm. Well, unfortunately, that's all the time we have. Thank you again for taking the time, and thank you everyone for listening.